thank you, Josh, and everyone for giving this opportunity for this talk. So, in addition, just in just bioprinting the tissues, you're also looking a lot into the assessment of these tissues. How do you sort of look at the characterization of these tissues? So I see some of the key team members, and I'll talk about them at the end too. So we are interested in the space domain. So we know that when you are exposed to microgravity and space radiation, there is vascular dysfunction. And the integrity of the smooth muscle cells and the endothelial cells are compromised. So how do you and there is a little bit of information to assess them right now. There is some work going on. In fact, I was looking at an interesting article which has been published that they analyzed the astronauts who were in the Apollo mission. There were 5 times the death compared to that of the low orbit, which is, or to the astronauts who were non-astronauts. So that was very interesting, fascinating data, which sort of was a motivation sort of continuing this research. So endothelial stress is one of the important aspect which is present during gravitational unloading, and also for space radiation part, the heavy ions are also very important element which needs to be analyzed. There's a lot of work, of course, with the X-rays and gammas, but these heavy ions are significantly different compared to what you see in X-rays and gammas. So 3D bioprinted tissue we think will be an ideal model to sort of look at vascular dysfunction when you're exposed to microgravity and space radiation. So the key component here is the vascular dysfunction, which happens in a damaged endothelial. So endothelial is one of the key component in maintaining vascular homeostasis. So this is a sort of a busy slide, but this is sort of we are looking at. So we are looking at the nitric oxide as a key measure of our cardiovascular function. And if you are sort of familiar with it, The NO is sort of a major endothelial dependent vasodilator which is present in the body. It helps actually to also regulate the blood pressure and also proliferation of the smooth muscle cells. So it's an important component in protection against the onset and progression of CVD. So we are looking at the response of bioprinted tissue to nitric oxide. And also in our body, the renin-angiotensin system, that actually produces angiotensin 2, and angiotensin 2 and NO has sort of competing effects. So there's a reduction of NO production due to availability due to angiotensin 2, and that will actually affect your cardiovascular response. So again, in microgravity or even in also space radiation, we are looking at the oxidative stress. And of course, we know that with oxidative stress, your ROS is going to increase and your availability of NO is going to decrease. So we are focusing on both, but right now we are starting with the nitric oxide because upregulation of angiotensin 2 will cause a problem in endothelial function because of the decreased NO level. So these are some of our broad objectives, sort of immediate short term. We are printing cells, the bioprinted tissue with HUVEC endothelial cells, and we are doing a lot of similar to microgravity. We are actually making use of local KC. They have a whole new array of microgravity simulators which are helping us to do a lot of analysis which we are sort of focusing on right now. And of course, the next goal can be make use of hopefully the ISS, NL, and other important part is that we are using tools, developing tools, a biochip, microfluidic biochip, which I will show in the next few slides for real-time imaging and omics analysis. And this will of course help us to study vascular disease and potential therapeutic drugs. So there is another application which is we are also working on. Yesterday, actually, Dr. Roberts saw a very nice slide that how the cells grow differently in microgravity versus in Earth. And one of the things which happens, and this is something we have been working with, I guess I'm hitting the wrong, with Mayo Clinic, that they're interested in liver cancer therapy, and one of the main component there is the lack of stroma which you develops in a cell culture in the earth. So if you can make use of the microgravity, we think that has give us significant, interesting results. So that's something we just started and work in progress. So we have been using a commercial 3D bioprinter and working under the hood. But since a lot of our focus is also in the space applications, actually we started this about a year and a half ago and developed sort of in-house. So we are sort of doing a lot of comparative studies with the commercial 3D printing and also an in-house. Actually, this started as an undergraduate student design project, and actually we are one of the 6 finalists last year in the BMS annual meeting for all scenario design projects in the BME biomedical engineering field, and we are one of the top 6 finalists. So it is a very low cost, homegrown, 3D bioprinting, extrusion-based 3D bioprinting system. Has got the basic, features. We can have a temperature control, humidity control, and pH and the UV light, so that we can sterilize it. So we have been sort of doing a lot of comparison of a commercial 3D printing onto the biohood to that of our self-contained bioprinting system. And it, of course, incorporates the laminar flow system, which is very advantageous for preventing contamination. And of course, we also have a good, strong, solid, everybody's doing the same thing, solid modeling. So we have a strong background team. We are using PT Creo and then slicer models to do that. And we're using also sacrificial bioprinting technique to print the channels. And we can, by sacrificing the channels, perfusion channels, we can sort of fill them with the endothelial cells. And so that's one of the techniques we're using in our printing, and it's a great tool because we can print. We have been printing a lot of perfusible vascular architecture using the sacrificial bioprinting technique. So some of the other parameters. So this is the one we have been starting right now. We have been actually getting a lot of data right now. We are using the simulated microgravity. There are many systems at KSC. One of them is the Gravite system, and which has rotating frames, and we can do a lot of simulated microgravity with our bioprinted tissue samples right now. And the other thing we're working on is with a company to develop a microfluidic live cell imaging for our bioprinted tissue. And the idea is that we can actually get a real-time, for days and hours data of a bioprinted tissue, and we can come back after they're done, come back either frozen or room temperature, depending upon the length of the ISS missions, and do a lot of omics analysis. So this is like a 4-well configuration, and they've been using hollow fibers, so that actually that's very advantageous, so that you can reduce the shear stress actually on the cells while you're monitoring them. And this can be adaptable to a wide range of cells, and this hollow fiber combinations configuration is very helpful from that aspect. And we can produce different types of drugs, and one of the things I'm going to show you also in my last slide towards the end is that one of the things we are also doing that we are trying to develop to prevent the angiotensin II receptor. We are trying to use angiotensin receptor blocker so that you can prevent cardiovascular disease. So this sort of thing will be very helpful because we can put different types of drugs in parallel and do a lot of studies with the concentration with different types using this 4-well chip hollow fiber configuration. So this has got different, you can do a lot of phase contrast and fluorescent microscopy imaging. It has got different objective lens, 7 different wavelengths. So this system we are testing right now in a simulated microgravity environment. And also, we also have an idea of we have been also using a bioreactor system, which is very standard. So our focus has been on the characterization part, and everybody in this field is sort of doing the same thing. Everybody starts with doing rheological analysis, then do live dead, and then these are our sort of focus right now is the nitric oxide and the myograph in the histology, and we're going to, of course, continue with the immuno and PCR for the omics part. So we always have to look for the bioinks, the shear thinning properties, so use rheological studies to sort of make sure that your bioink is, we have been using alginate, gelatin, fibrinogen, and different types of recipes to sort of check our bioink composition and their shear thinning properties. And then we do live dead. Right now we have looked at 7 days, and you could see at images of day 4 and day 7 using a standard, calcine and ethidium homodimer live dead assay. And this shows that after 7 days we have a nice, still healthy green cells in our bioprinted constructs. And we also do MTT. One of the reasons we did MTP, the MTT is that we want to sort of optimize our alginate gelatin concentration, because MTT will give you the metabolic assay. So we have been trying to optimize it. This was some sample one, that how do you optimize the different alginate gelatin concentrations, which people have actually been used, and try to see that what may best fit in terms of viability for our applications. So this is sort of our key focus right now, is looking at the nitric oxide release from tissue constructs from HUVEX. And one of the things when we started looking into it, we didn't really see any paper actually on nitric oxide on bioprinted tissue. So we wanted to make sure that the bioprinted tissue response, the same protocol which has been using animal tissues for NO, sort of will be applicable. So what we did initially, we used two reagents, LNAME and SNP. One is a nitric oxide inhibitor and one is the nitric oxide enhancer, just to ensure that the response you're going to get is what you're going to expect. So we did quite a few significant studies before we working on the simulated part. So, and we got expected results. NLAME is an agent, if you're familiar with it, sort of decreases your nitric oxide production, and sodium nitroprusside sort of increases your nitric oxide level. So with a bioprinted tissue, we got very consistent result, which we wanted to sort of do before working on the microgravity, simulated microgravity. So again, in a quantitative fashion, you could see this is a control, and SNP, which should see an increase in nitric oxide level and decrease, because this is very significant, because nitric oxide level is directly proportional to onset of CVD, cardiovascular disease. So, and then we sort of started using the simulated microgravity environment at KSC. Again, we put a lot of control samples and then simulated microgravity. I'm going to show you one or two sample data. So this is what we observed. And these were kept in the microgravity simulator only for a couple of days. So what we have been actually looking at is also finding out that there are some studies that showing that due to the autoregulatory mechanisms, that maybe your NO production will sort of stabilize or increase or decrease. So we want to actually now extend this to do this, keeping the simulators for 14 to 21 days, like ISS missions, and see the response. So this is after 48 hours for two sets of experiments, and you could clearly see, compared to the control, you have a significant reduction in the nitric oxide, which sort of will correlate with the CVD. So, and also we're looking at myography right now for the relaxation and contraction of the muscle functions. And we are able to also get interesting results as a starting point of the characterization part using the bioprinted tissue. And then the histology, which is also part of the requirement, also the vascular tissue challenge. So we have been trying to look at our matrix and look at our tissue for the histology. We have not much made some progress. We have some very preliminary data. So these are some adapted data showing that what you can expect from the immunochemistry and the PCR and RNA, mRNA analysis. So it's work in progress. So to, again, so what is our immediate goal is to, of course, be part of the vascular challenge, able to print thick tissues, perform histological studies, and do a lot of this biochemical and function analysis. And as I mentioned, that we want to really keep this samples in simulated microgravity for an extended period of time, try to see like a time domain map of the nitric oxide release and the ROS, the oxidative stress, so that you can correlate that. And this is another also key of our focus area. We actually want to use angiotensin receptor blocker. Losartan, for example, is one such example. We want to just see if you can restore the cardiovascular function. If you sort of pre-treat the tissues with losartan, does you restore the function? So we are doing a whole bunch of planning experiments with simulated microgravity to look into that. And of course, our long-term goal, of course, is to do real-time imaging of NO and the ROS. Hopefully able to use the ISS and we are also planning to make use of the Space Radiation Laboratory, which has got the simulation for the radiation. So I want to thank, so myself, we have a vascular biologist, we're using KSC, we're using H&E Photonics, David for the life cell, and these are some of the students we are working on. Okay, thank you for your time. I answered everything. Okay, thank you again.